Hello everyone and welcome to our webinar of today. Our speaker is Mattia Uzreli from the ETH Zurich, who will be telling us about the characterization of gels using dynamic light scattering, starting with an introduction to the concept of non-ergodicity and also including an example linked to the characterization of amyloid fibrils. So let me hand it over to Mattia for the main presentation.
So many thanks, Colleen, for this very kind introduction, and welcome everyone to this webinar with title Characterizing Gels with Dynamic Light Scattering. So the structure of the presentation is the following. First, we will talk about dynamic light scattering, so how the technique works and how we should treat the data. Then we will dive into non-ergodicity. What is the definition of non-ergodicity, the differences between time and ensemble average measurements, and which is the influence of this on dynamic light scattering measurements. Then we will talk about an application more in detail, how we could use dynamic light scattering coupled with non-ergodicity to measure the mesh size of amyloid fibril gels. To see how this let's say, ensemble of dynamic light scattering and non-ergodicity actually allows us to extract a structural parameter of amyloid fibrils gels that is the mesh size. So, dynamic light scattering. Here we are. This is the classical configuration of a dynamic light scattering experiments. What we have an incoming laser beam that you can see on the left, and then afterwards, at the specific angle theta that, for example, with the LS spectrometer can change, or in other equipments can be fixed, it depends. What we do is that we put a lens, we put a photon detector, and we have a look at how the intensity at the specific speckle of light in the far field changes over time. A very important thing to be discussed is that, for example, in the LS spectrometer, a very nice thing that could be done is to change the measurement angle. What happens when I change the measurement angle? If I increase the measurement angle, I increase a quantity that is called the Q vector, and the absolute value of the Q vector is actually telling me something about the characteristic length scale that I am proving. So keep in mind that when I do a dynamic light scattering experiments, I increase the measurement angle, I increase the Q vector, and I decrease the characteristic length scale at which I'm having a look at the phenomena that are relevant for my sample of interest. So it means that when I increase the angle, I'm having a look at the smaller and smaller and smaller length scales. From a pragmatic perspective, what do we do? So, as we mentioned, we detect the intensity of a single speckle of light, for example, with an optical fiber, we send it to the correlator. What we see actually is that the intensity fluctuates in time. If we define an intensity correlation function, so basically we have a look at how much the intensity is self-similar to a translated version of itself of a characteristic time tau, we compute this quantity, we obtain from a dynamic light scattering experiment something that looks like the curve that we have on the right. So this is the quantity that we measure. It means this is the empirical data that we get from the machine. Afterwards, to make an interpretation of this data, we actually need a model. We need to describe it and we need to treat it. So the usual way this is done is that we go from the intensity correlation function through the Siegert relation to the intermediate scattering function that is related to how the scattered electric fields like is kind of interacting with itself. So from the intermediate scattering function, in the traditional dynamic light scattering experiments, we can extract the diffusion coefficient or information about the motion of the objects in our system. So once we have extracted the intermediate scattering function, we can have different choices. For example, let's imagine that we have a monodispersed system of Brownian particles. In this case, our intermediate scattering function can be defined by a single exponential decay that depends on time, depends on the Q vector, but you see the quantity D that is placed inside there. This quantity D is the diffusion coefficient. Then through this relationship that contains the absolute temperature, the viscosity of the medium, and the diffusion coefficient itself, I can extract the characteristic size of the objects that are diffusing inside my suspension. Let's imagine instead that we do not have a monodispersed system, but we have a polydispersed system of Brownian particles, but that still they have, let's say, some polydispersity around a single population. In this case, for example, we can use the cumulant analysis where we do a viral expansion of the expression that you see above. And here we can extract not only the average diffusion coefficient of our particles, but we can also see actually a deviation of the diffusion coefficient itself that is telling us something about the polydispersity itself of our Brownian suspension. Let's now imagine that we have a polydispersed system of Brownian particles in this time with multiple populations. So it means that I do not have a single exponential decay that is actually a very broader, but I really have multiple exponential decays. In this case, we can use, for example, a technique that is around since the 80s of last century. This is the Contin analysis. And actually, a LES instrument has developed a very robust and very, very nice algorithm that was like arising from many years of work that is able to extract the size distribution in a very, very accurate way. So now we've been talking about Brownian suspension. It means that if I put myself in the shoes of a particle that is inside the suspension, what is happening is that I'm simply kicked by different solvent molecules, and so I am performing a random walk around. But there is nothing that is preventing me as a particle to move around. I can move freely. I'm simply kicked around by the solvent molecules. Actually, there are systems in which this is not true anymore. So let's have a look at this situation here. What you see on the left is actually a system of Brownian objects. You see that they can move very nicely and they do not interact that much with each other. But what you see on the right, actually, you see some particles that are embedded, for example, in this case, in a network. So here we have different contributors. What happens? That are still the solvent molecules that are hitting the particles that they would like to move. But at the same time, they're kind of constrained by some forces of the network in which they are embedded, and they cannot move freely as they would like to do. So which are these intuitive differences that we're talking about? On the left, particles are free to move. On the right, particles are not free to move because there are also some other energetic components that are keeping them in a kind of a more fixed position with limited Brownian excursions around this position. At the same time, another very, very important aspect, if we have a look on the left, we can say that subvolumes of the sample are similar to each other and carry the same pieces of information of the entire sample itself. But if we have a look on the right, it is true that some particle, if let's say the surrounding network is similar, has similar dynamics. But if I take a part of the sample, maybe in that specific part of the sample, the particles are embedded in a specific way. If I take another area of the sample, the particles are embedded in a bit different way. So actually the subvolumes of the sample itself are not entirely self-similar to each other. They have some difference, especially in how these particles are placed compared to each other. So what is non-ergodicity? These different areas of the samples that, let's say, have different absolute configuration and different behavior, sometimes there are different sub-ensembles. So let's imagine that we have the sub-ensemble 1, the sub-ensemble 2 that have no correlation among each other. Usually, what happens, what we do in typical dynamic light scattering measurements or when the ergodic hypothesis is holding, is that I can go from the sub-ensemble 1 to the sub-ensemble 2 that have no correlation, simply waiting. It means that the system has time to explore a bit its phase diagram, it will move around and will explore the different sub-ensembles. But when I have non-ergodicity, what happens is that time is not enough. So it means that time is actually not enough to jump from one sub-ensemble to the other. So this makes some points arise. The first one is that a time average measurement is allowing us to prove only a single sub-ensemble. It means I'm not proving all the configuration of the sample, all the possibilities of the sample itself, and simply proving what the sample is doing in that specific volume that I am considering. The questions that are arising from here are, how can we extract information on the entire sample? So it means the full ensemble of possibilities. And the second question that is actually very interesting is it actually possible to extract information on the entire sample starting from a single subensemble? Because we said that the dynamics, if the sample at least is homogeneous, should still be the same. So is it possible to do this? So this is what we saw before. This is a typical dynamic light scattering experiments. We have our intensity as a function of time. We perform an intensity correlation function. That is our, let's say, measurement data. We go from this to the intermediate scattering function through the Siegert relation, and we extract the diffusion coefficient. Our question mark is, now non-ergodicity is jumping in. Does this equation still hold true? So the consideration that I will share from this slide on are present in this very nice article that I put here from Puzy and Van Meegen, that I think is one of the most important articles for DLS and non-ergodicity. That is actually deriving the equations for treating non-ergodicity properly. So this is the question that we were wondering. Does the usual data treatment, the Siegert relation, still hold? Can we still divide the data for beta and then take the square root to have our intermediate scattering function? Actually, it's a bit tricky because we mentioned before that time is not enough anymore to obtain ensemble average quantities. So at this stage, when we deal with non-ergodicity, we need to take into consideration both time average quantities, so time average intensity correlation function, and ensemble average intensity correlation function. So what is the difference? The one on the left is the classical dynamic light scattering experiment that I am performing. I put the sample inside the machine, I measure a specific speckle of light, I take the intensity correlation function, and that's it. So I'm measuring a specific location of the sample. What you see on the right, the ensemble average intensity correlation function, as you will see also later on, is actually a quantity that is telling us, yes, how the intensity is correlated to itself, not anymore in a single subensemble. I will say now I consider this subensemble, then another one, I make an average among all of them, and now here I am with the properties of the entire sample. When I have a Brownian sample, when I have an ergodic sample, the two quantities merge. With a time average, I can measure an ensemble average. Non-ergodicity, the ergodic hypothesis is not true anymore. I have to consider them separately. So actually, in the article of Puzy and Van Meegen, they're showing what's going on to the time averaged intensity correlation function and to the ensemble average time correlation function. So on the left, you see the time average. On the right, you see the ensemble average. What are the differences? So first of all, they are not the same. We can see from a visual appearance that they do not much look the same. First of all, we have a different behavior of the intercept. And this is very, 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 very important that we will also see later on. The ensemble average quantity starts as usual near to 2, or when we take the G2 minus 1, it starts near to 1, where it's not sometimes maybe the difference between the actual value and 1, it's simply related to the specific design of the machine. But actually, if you have a look on the left, here we see 3 different curves that are all from the same sample, but they have different intercepts. So it means that when I do a measurement on a non-ergodic sample, actually I can have different intercepts considering different subensembles. Also, we see a different behavior of the plateau. So when I do a time average intensity correlation function, G2 decays to 1, so it means I take G2 minus 1, it decays to 0. But actually, the ensemble average intensity correlation function is telling me something different, that my function is not decaying to 0, and this is related to the fact that actually my colloidal objects cannot perform a full displacement compared to the characteristic length scale that I am analyzing with my dynamic light scattering experiment. So How to recognize non-ergodicity and what to do. What you see here in this graph are 4 measurements. You see in the green color the ergodic sample that you see, very nice. It starts from around 0.9. So this is the intensity correlation function. It's close to 1. It's very good. And it has a full decay. Then I put inside my machine a non-ergodic sample. I rotate it so that I prove different sub-ensembles, and I take 3 different measurements. And you can see here in circle, in squares, and in triangles of different colors, the different measurements that I get. So I confess, when I was doing these experiments, I was thinking, okay, either there is something wrong with my sample or there is something wrong with the spectrometer. I have to call for doing a realignment. There is something wrong. Actually, the answer is no. It's normal that when you are measuring a non-ergodic sample, actually the intercept is dropping quite a bit. So you see here the intercept are different. So the intercept of non-ergodic sample are typically lower than the one of the ergodic samples, and we measure different sub-ensembles. Also, the intercept is changing. So first point, very, very pragmatic point. You take your sample, you put it inside the machine, you make a measurement, you see that usually for most of the sample that you measure, the intercept is close to 0.91. For this specific sample, you see that your intercept is lower. Maybe it's around 0.6, maybe it's around 0.4. You say, what's going on? I don't understand. I suggest a little thing. Try to move or to rotate the sample. Try simply to check what's going on in another sub-ensemble. If you see that the intercept is changing, there are very high chances that you're dealing with non-ergodicity. So this means that you can still perform the experiments. You can still collect very, very insightful information of your sample. At the same time, you need to treat the data properly. So our question now, we saw that for usual Brownian motion, we use the Siegert relation. What shall we do with non-ergodic samples? So what do we do? Let's ask a scattering expert, that in our case is Homer Simpson. Maybe Homer Simpson can suggest us, yeah, who cares if the system is non-ergodic? The Siegert relation is a very strong one. It can be applied in many, many contexts. So let's simply apply it. The intercept is lower, but still, if I rescale with the value of beta, what is the problem? Then I have an intermediate scattering function that is still going from 1 to 0. No problem at all. Let's use it. Let's see what happens. We do it for the non-ergodic samples, and we see that the curves actually are overlapping quite nicely, and they look like the usual curves that we extract from the typical dynamic light scattering experiments. Our scattering expert is very happy. However, he should not be that happy. Why? For this reason, if you have a look at the article of Puzy van Megen, they explicitly say that actually the diffusion coefficient that you extract from these curves that you see on the right are only apparent diffusion coefficient, because to get the true diffusion coefficient of your system, you need to apply a correction that depends on the intercept of the initial intensity correlation function, and depends a bit on how differently your specific sub-ensemble is scattering compared to the entire ensemble. So here, if we want to trust the diffusion coefficient, we will need to apply a correction. Moreover, something that we observe is that the curves decay to 0. That is not consistent with the fact that our Brownian objects are performing only restricted motions. And so, as we saw before in the treatment from Puzy and Van Megen, they should not perform an entire decorrelation of the function. So if we apply the Siegert relation to the data that we extract from non-ergodic samples, we have that the motion of the colloids is generally overestimated, both in absolute quantity and also in terms of diffusion coefficient. Our scattering expert is a bit lost. What shall we do then? Here we are, how to treat the non-ergodic data. So first point, before we were asking 2 different questions. The first question was, how do we extract information on the entire sample, the ensemble itself? Actually, the first method that we can think about is the brute force method. We can say, okay, sample, you are non-ergodic, I'm going to deal with you properly. It means that I make a measurement in one specific location. I make a measurement in another location. I go on, I take many, many measurements in different sub-ensembles. Then if I have been mapping properly a huge number of sub-ensembles, I perform an average and I can extract the ensemble average, the time correlation function, intensity correlation function, sorry, that is also time but performed on many sub-ensembles. This method works. The thing is that it takes time because I really have to prove different sub-ensembles. Then I have to check one, I have to rotate the sample, I have to rotate the samples many times to understand what's going on. Other methods are, for example, ecodynamic light scattering or using multispectral dynamic light scattering. It is already performing an ensemble averaging by not considering a single speckle of light, but by considering many of them. And I will redirect you to this very nice book from Otto Glatter in the chapter on dynamic light scattering for having a look a bit more about how these methods work. The second question that we asked was the following. Is it possible to extract information of the entire sample, it means the entire ensemble, from a single sub-ensemble? Because if I manage to do this, I'm much faster. It means that I need simply to measure a single sub-ensemble, treat the data properly, and then I have the information that are actually telling me something about my entire sample. So how does it work very practically? So here I want to share a recipe. So take paper, take a pen, write down. What I personally suggest you to do when you're dealing with a non-ergodic sample is the following. You go to the equipment, you turn it on, you wait that the laser is heating up, everything. You perform the usual measurement as in dynamic light scattering. One is potentially enough. If you want to have more statistic, also to see if maybe your sample can be a bit heterogeneous in different sub-ensembles, you perform up to n measurements. So perform the measurements as usual. Here it is an example. So here on the left, you see three typical intensity correlation function from a non-ergodic samples. So what are the quantities that you should be interested in? One, the central one, is the intensity correlation function that the software of the equipment is giving you directly. The other two quantities that you should be interested in are actually the reduced intercept of the function. You see here, for example, in these three curves, we have one that has an intercept that is close to 0.1, one to 0.15, one to 0.2. These values are important for the correction afterwards. And we are also interested in having a look at how that specific sub-ensemble in average is scattering. So when you perform a dynamic light scattering experiments, the software is actually giving you back the count rate. So what you're also interested in is for each measurement, having a look at what is the average count rate that you're measuring in that specific measurement. So up to now, nothing new. These are all things that the software is directly giving you. The software is giving you the intensity correlation function, is giving you the value of the intercept, and is also giving you the value of the average scattered intensity. Afterwards, we do the following. We do an experiment where we prove the so-called ensemble average scattered intensity. It means that here we are not having a look at anymore at the dynamic information. We are not having a look at the correlation function. The parameter we are interested in is the average scattered intensity that the system is measuring while the sample is being moved or rotated. So basically, I perform a long measurement over which I rotate the sample at a constant speed in order to prove different sub-ensembles, and I detect how the average scattered intensity looks like while I'm doing this rotation. Afterwards, when I perform my n measurements in specific location and I perform my ensemble average measurements to detect the average scattered intensity, For each measurement, I can take the ratio between the ensemble average scattered intensity and the time average scattered intensity of that specific measurement. And afterwards, I can use this quantity as a parameter together with the reduced intercept to correct the time averaged intensity correlation function that I collected in the first n measurements. I correct each of them using the same protocol. So now in the graph on the left, again, you see three different measurements. For these three different measurements, everyone is characterized by different values of epsilon, because the ensemble average scattered intensity is always the same, but the time average scattered intensity changes for each sub-ensemble. Everyone has a different reduced intercept. I applied this formula to everyone, and actually you see that three curves that in the beginning look different to each other, now they are nicely overlapping. It means that the motion of the units that I'm proving is the same. And finally, I observe a quantity that is starting from 1 and is actually decaying not to 0, but to a non-ergodic plateau value. Now let's see an application. So in my personal research, I've been applying DLS and non-ergodicity to amyloid fibrils, but let's understand a bit more what these amyloid fibrils are. So what you see here is the protein energy landscape. So you know that proteins are actually very functional units that are used in biology to perform so many tasks, for example, enzymatic reaction or to build structure in the body, many things. What you see here is that actually each protein is characterized by a sequence of amino acids. So if we take a protein in the unfolded state, it's simply a sequence of amino acids, but due to the specific interaction among these amino acids, the protein can actually either fold into the native state, that is the one that is giving the functionality of the protein itself. But if this process does not go very nicely, or if there are other external conditions that are unfolding the protein and are driving some processes, we can actually also form these amyloid fibers that, as you can see here, actually represent a minimum in the protein energy landscape. It means that the oligomers and the different amino acids place themselves in a configuration that is very, very stable. So more in detail, we have the formation of this cross beta structure that actually afterwards is forming single protofilaments that are laterally aggregating to form some mature amyloid fibrils. In this review article, you can actually see that amyloid fibrils can be, for example, pathological. For example, the formation of amyloid plaques in the body is associated to different diseases, for example, as Alzheimer, as Parkinson. They can actually be functional. For example, bacteria are employing functional amyloids for motion or for storing purposes, but they can also be artificial. It means that we can start from protein, we can denaturate them, we can drive in vitro the synthesis of artificial amyloid fibers that can be used as starting materials for innovative applications. For example, if you start from a very famous protein, this beta-lactoglobulin, that is the main protein fraction of whey, which is a residue from cheese production, we can see that if we form a suspension that is at 2.8% at pH 2, and we heat it up at 90 degrees while stirring for around 5 hours, we form these elongated amyloid fibers that you can appreciate in this AFM image. These amyloid fibers actually, on the side of being very, very interesting from a theoretical perspective, they can be used, for example, for universal water purification because they absorb very, very nicely heavy metals, or they've also been using, for example, for iron ore fortification. So it is extremely important to understand the properties of the physical states that these amyloid fibrils are forming. From a polymer physics perspective, you can think a single amyloid fibrils as a semi-flexible polymer. It means that it has like contour ondulation on a length scale that is similar to the length of the fiber itself. And actually, if we are at pH 2, these objects are positively charged. If If we take enough amyloid fibers, and as they have a very, very high aspect ratio, we don't even need to go so high in concentration, these amyloid fibers are forming a network. So it means that we have an average distance between the different amyloids that is called the mesh size, and the amyloid fibers themselves are meeting themselves in some points that are called entanglement points. If we have a closer look here, what happens? The fibrils are positively charged, so when they get in contact with each other, actually they do not really manage to get in contact because we have some electrostatic repulsion. It means I have an electrostatic force that is pulling the fibrils apart. So it means it's an entanglement point. The fibers can kind of dance around, but they don't manage to stick on each other. Something that I can do is actually to increase the ionic strength. You can see here in the presentation that I put more ions around so that I screen the surface charge. The other fibril now, from a far away perspective, does not know anymore that the fibril is charged, so manages to get near, and it gets to a point where we have the van der Waals forces, so the dipole forces, actually manage to pull the fibers together and to transform the entanglement points that make the status of the suspension as a viscous sol into an elastic gel, because these physical crosslinks become long-lived. What was the research question that inspired this work? So here is our amyloid network with a specific value of the mesh size. Actually, there is also a very important quantity that is called the entanglement length. That is basically the distance that I have to travel on a single fibril between two different entanglement points. We see that if we add salt to the system, we transform these entanglement points into long-lived physical crosslinks. And actually, what happens? Let's imagine now, let's dream about being an amyloid fiber in solution, okay? Let's imagine that I am there by myself. I am not surrounded by other amyloid fibers. What can I do? I can do different typology of motion. For example, I can diffuse, I can rotate, and actually, for the fact that I have a very, very high aspect ratio, I can also perform ondulations on the contour. So now let's imagine what happens, me as a single amyloid fibril, when I'm meeting other amyloid fibers. Okay, I start getting in contact with these other amyloid fibers in different entanglement points. I think you agree with me that intuitively, I cannot bend as before. For example, if a bending mode is suggesting me to bend along my entire contour, I cannot anymore because now I have two fixed points in which I am in contact with other amyloid fibers. The other amyloid fibers are hindering the bending modes of me single amyloid fibers. You can also imagine the following, that the more amyloid fibers I put in the system, it means the higher the concentration that I'm considering, the more the bending modes are hindered, because basically the less I can express my fluctuations. Therefore, the question is the following. Is it possible to use dynamic light scattering together with the non-ergodicity treatment? Because we saw when we add salt, we transform the entanglement into crosslinks. We have a gel that is non-ergodic. Can we measure XEM, that is the mesh size, as a function of concentration by proving how much the bending modes themselves are hindered? So, what did we do to perform this study? We used the 3D less spectrometer that we have here in our group of food and soft materials at ETH Zurich. So LS instrument is providing this very, very nice equipment that can perform a multitude of experiments. It's an advanced light scattering equipment that is suitable for performing the following characterization. I can, for example, check the size of nanoparticles, prove the properties of polymers, peptides, proteins, emulsions, and also extracting a huge range of information. If there is the 3D modulated technology, we can also prove concentrated system to extract the particle size, the particle shape, molecular weight, aggregation state, and it can also prove the gels. We mentioned before that to perform the ensemble average measurements, we need to rotate the sample. Well, LS instrument for the LS spectrometer is actually providing the sample goniometer. You can see in this video here, actually, that it is possible to set the sample goniometer to perform a constant rotation of the sample with a specific speed, so that I can also perform my ensemble averaged intensity that is scattered from the sample, that is then useful to correct the single measurements that I perform when the sample goniometer is kept in a fixed position. So we use the 3DLS spectrometer. What did we do? Here is a topology of measurements that we acquired. So what do we see? Here you see three different curves. You see the red circles, the blue squares, and the orange triangles. These are different measurements at different concentration of amyloid fibrils. What do we see? So when the concentration of amyloid fibrils is increased, the plateau of the intermediate scattering function is also increased. Why? For what we said before, from an intuitive perspective, the concentration is getting higher. Due to the fact that the concentration is getting higher, the fibrils are more next to each other, so the mesh size is getting lower. The entanglement length is getting smaller, and this means that I have only small portions of my fibrils that can perform transverse oscillation. So it means the motion gets more and more constrained. Something also interesting that you can see from this data is that the higher the concentration, typically the smaller the standard deviation of the intermediate scattering function. This is related to the fact that the higher the concentration, the more the sample is getting homogeneous. So also the single sub-ensembles that I'm proving are actually more similar to each other. This is an intuitive explanation, but if you want to extract quantitatively the value of the mesh size, we also have to have a physical explanation. So from a physical perspective, this is the formula that we mentioned before for non-ergodicity. This needs to be coupled together with a theoretical treatment of the intermediate scattering function from semi-flexible polymers. So here, this very, very nice treatment from Croy and Frey is telling us that if we consider times that are larger than the characteristic bending time of a fraction of a fibril that is as long as the entanglement length, my function, my intermediate scattering function, is decaying to a value that is related to the mesh size of the sample itself. For a more precise derivation of this equation, you can have a look at the reference below. That is the paper that we published in our group out of this work in macromolecules. And actually, we put everything together, and we see that from a dynamic like scattering measurements, we can extract a static parameter of our sample. So what you see in this slide, in this graph, is the value of the mesh size. It was extracted as a function of concentration. More in detail, you can see measurements at two different angles. Later, I will explain you why to perform measurements at different angles and which different informations we can get. You can see that actually the experimental data that are the red circles, blue squares, and orange triangles are in agreement with a theoretical estimate that we made independently with a modified cubic lattice model. So there is a good agreement, and the data are also nicely following the expected scaling law of the mesh size as a function of concentration, that is with the power of -1/2. But I just mentioned, here we measure at two different angles. Why? And more in detail, what is the role of the measurement angle? Here we again go to a very practical aspect that I think it's extremely important when performing measurements in DLS. So before we were keeping the angle fixed in the graph before and changing the concentration. Now let's try to do the opposite. It means we keep the concentration of amyloid fibrils fixed and we change the angle at which we measure. So for example, here you can see that we go from 60 to 135 degrees. And let's keep in mind what we were saying in the beginning of this presentation. It means that when I increase the angle, I'm increasing the Q-vector, and by increasing the Q-vector, I'm having a look at smaller length scale. So the system is the same. It's still 0.38 milligram over milliliter. The absolute displacement of the fibrils is the same. But what happens when I increase the angle? I'm having a look at smaller length scale, so it is reasonable that the same absolute displacement observed over the perspective of smaller length scale, it seems to be the correlating more. I have a lower non-ergodicity plateau value. So that's why by increasing the angle, you see that the intercept value is getting down. So again, we do our measurement. This is what I just said. This is related to the fact that we change the angle, we change the length scale. But actually here we see something puzzling because we say, okay, the different length scale that I'm considering are related to the different wave vector. Actually, the formula for extracting the mesh size contains the wave vector. So once I correct for the wave vector, it's okay. I should be able to extract always the same mesh size. But this is actually not true because we always have to remember that when we perform a dynamic light scattering experiment, actually we are having a look at the relevant motions at the length scale that we are proving. For example, let's imagine that I am having a look at the crowd of people. Like, for example, if I am in the crowd and I follow the motion of a single person, maybe this single person has a specific behavior. But now let's imagine that I go on an helicopter. I have a look not at the specific zone of the square, but I have a look at the entire square. And maybe I see patterns of people moving that by having a look at the single person, I could not see. The same is with dynamic light scattering. So what happens here? Here is an example of the mesh that we are considering. When we are at 60 degrees, so basically the red point, the circle red point in the graph on the right, we are not having a look at the single filament, but maybe we are a bit larger than the mesh size itself. So we are having a look at multi-filament dynamics, which is not maybe only binding fluctuations or bending fluctuations, sorry, of the single fiber itself, but maybe they have some cooperative motions that are making the correlation function decay more, and so I have an overestimation of the mesh size. At the same time, when I increase the Q-vector, you see here that by increasing the Q-vector, we have a look at smaller length scale until we approach the characteristic size of the mesh size. So I am having a look only at the single fraction of a single amyloid fibril. In this case, the value of the mesh size that I am extracting becomes more and more reliable because actually the theoretical model that I've been using that describes the behavior of the single amyloid fibril is aligned with the length scale that I am proving. So two very practical suggestions. If you want to extract information of non-ergodic samples, on the one hand, use the correction protocol that has been shown in this presentation. And on the other hand, I think it is very, very important to compare which is the characteristic length scale that is proved by the wave vector at which you are doing the measurements, and which is the characteristic length scale inside your sample that you want to extract. And the more they are comparable to each other, also especially in light of the theoretical model that are used to extract information, the better it is. So, quick summary, because I think I was throwing really a lot of information, and so I'm really looking forward to your questions later. We saw non-ergodicity, we saw the definition of non-ergodicity, time versus ensemble, and we saw a brief recap of dynamic light scattering, the technique, the influence of non-ergodicity, and how to treat the data properly. Afterwards, we saw an application on how to combine non-ergodicity and DLS to extract the mesh size of amyloid fibril gels with a rigorous theoretical treatment. So many, many thanks. I would like to thank all the people in the Laboratory of Food and Soft Materials, especially all the ones that are co-authors and who have been helping me a lot actually in thinking about what's going on inside this system. Many thanks to LS Instruments, to Pauline, first of all, for the possibility of having this webinar, also for providing so nice equipment for doing light scattering experiments, and also for always being present when I have questions, how shall I do this? I did some bullshit, so maybe the spectrometer is not aligned anymore. Can you please come to align it? Always very, very available and looking forward to help us to put everything on track. And with this, I'm done, and I'm happy to answer your question and to discuss.